Hi everyone, welcome to another revision video. This video is based on the enzyme topic of biological molecules, based on spec point 3.1.4.2 of the AQA A-level biology spec. So let's get into this. First of all, let's look at some key definitions. So an enzyme is a globular protein that acts as a catalyst for a reaction involving a specific substrate by lowering activation energy. An active site is a functional region on an enzyme that is complementary to a substrate. A substrate is a molecule on which an enzyme reacts by fitting/binding into an active site, forming enzyme-substrate complexes. A saturation point, specifically in this enzyme's topic, refers to when all active sites are occupied/saturated/ enzyme is limiting the rate of reaction / there's a maximum number of enzyme substrate complexes. Denaturation is the irreversible structural change in the tertiary structure of an enzyme that makes it unable to catalyze due to the active site no longer being complementary to the substrate. Onto some more definitions. An inhibitor reduces the binding of a substrate to an active site, preventing the formation of enzyme-substrate complexes. The induced fit model is the way in which an enzyme alters the shape of its active site to be exactly complementary with the substrate to form enzyme-substrate complexes. And a lock and key model is the older theory of enzyme action, suggesting that the enzymes have a fixed shape active site that is always perfectly complementary to a specific substrates rather than having an adjustable active site. And onto some key terms. Competitive refers to when it binds to an active site, so has a similarity in shape to the substrate, so competes with the substrate for empty active sites. And non-competitive refers to binding to a site on the enzyme other than the active site, which is also referred to as the allosteric site, so it has different shape to the substrate, so does not compete with the substrate. Moving on to the main content of this topic. Models of enzyme action. There are two models of enzyme action. The lock and key model, which you mentioned in the definition, is the older model, and the induced fit model. If we were to compare these two models, the lock and key model, initially the tertiary structure of the active site is complementary to the substrate, whereas in the induced fit model, initially the tertiary structure of the active site is not complementary to the substrate. Again, for the lock and key model, the tertiary structure of the active site does not change when the substrate binds, whereas in the induced fit model, the tertiary structure of the active site changes when that substrate binds. And a similarity between both models of enzyme action is that enzyme substrate complexes do form. So more on the induced fit model. Here is the process of the induced fit model. An active site forms as an enzyme and substrate interacts. Initially, the tertiary structure of the active site is not complementary to the substrate. The enzyme's general shape changes in the presence of a substrate, such that the tertiary structure of the active site changes when the substrate binds. This places stress on the bonds, within the substrate, inducing catalysis. Due to the enzyme being flexible, it is able to mold itself around the substrate, and after the products are formed, the enzyme will resume into its original shape. And on the lock and key model, it is as defined at the beginning. And when you are referring to the lock and the lock here is the active site of the enzyme and the key is the substrate. And some limitations of the lock and key model include is that it suggests that enzymes are rigid, whereas other molecules can bind to the allosteric site. And it also suggests that all enzymes catalyze a single chemical reaction. More on enzymes. Each enzyme lowers the activation energy of a reaction it catalyzes. Enzymes catalyze a wide range of intracellular and extracellular reactions that determine structures and functions from cellular to whole organism level. So more on the specificity of enzymes. Each enzyme has a specific primary structure, which is the amino acid structure. We go more into this when we talk about the protein topic earlier in this topic. It folds in a particular way, forming a tertiary/secondary structure, producing particular shape of active site, and the shape of active site is complementary to that of the substrate. When we're looking on how the enzyme shape relates to its function, it has a specific 3D/2 tertiary structure / shape, and that means the substrate is complementary to the shape and the substrate is able to bind to the active site. Now let's look at the roles of certain structures within this topic. The role of active sites within enzyme controlled reactions include increasing rate of reaction by lowering the activation energy. The induced effect causes the active site of an enzyme to change shape, so enzyme substrate complexes cause stress on these bonds within the substrate. Okay, now onto the role of the enzyme substrate complexes. It increases the rate and is able to do this by reducing activation energy due to the stressing of bonds within the substrate. Moving on to inhibitors, there are two types of inhibitors, competitive and non-competitive. In competitive inhibitors, you see features such as it being able to bind to the active site, has a similar shape to the substrate, it does not distort the shape of the active site, and its inhibition can be overcome by increasing substrate concentration, whereas when we look at non-competitive inhibitors, it binds to a site on the enzyme other than the active site. It has a different shape to the substrate. It distorts the shape of the active site, and inhibition cannot be overcome by increasing substrate concentration. My goodness me. And similarities of both these inhibitors are that they are able to slow enzyme action. Moving on, let's look at factors affecting enzyme-controlled reactions. These include enzyme concentration, substrate concentration, concentration of competitive inhibitors, concentration of non-competitive inhibitors, pH, and temperature. Okay, moving on to the effect of substrate concentration on enzyme-controlled reactions. Increasing the concentration of substrates, which, let me first note, this notation with the square brackets is very common in A-level chemistry, but I appreciate not everyone's an A-level chemist. So the square bracket purely means concentration of. So if you ever see me use the square bracket notation in any of my videos, that's what I'm referring to, the concentration of whatever's inside the bracket, which here is substrate. So going back to the notes, increasing substrate concentration will increase the rate as there are more substrate molecules, there are more collisions, so there are more enzyme substrate complexes formed. The rate of reaction will increase until the concentration of enzyme becomes a limiting factor, where increasing substrate concentration will have no further effect on the rate. The enzyme is said to have reached the saturation point at this point. And as you can see by this really cute diagram, hopefully if you are on video you will be able to see it. If you're on Spotify, make sure you press the video button or hop onto YouTube to see it. But I just drew a little diagram showing how this would be represented on a graph, that you may see something similar within your exam. Moving on, let's look at the effect of enzyme concentration. Increasing enzyme concentration increases the number of available empty active sites for substrates to bind to. This enables more enzyme substrate complexes to form. The rate of reaction will increase until the substrate concentration becomes a limiting factor, where increasing enzyme concentration will have no further effect on the rate. And once again, I have this really cute diagram, so hop onto the video if you can't see it. Let's move on to the effect of competitive inhibitors. This decreases the rate of reaction of enzyme-controlled reactions because the inhibitor has a similar shape to the substrate. Make sure to use the word similar instead of same here. There's just a little mark scheme thing. So it's able to fit / bind to the active site, which prevents enzyme substrate complexes from forming. And once again, there's a cute little graph, so you'll be able to see. Moving on to the effect of non-competitive inhibitors, this decreases the rate as it's able to attach the enzyme at the allosteric site, which we mentioned before is a site on the enzyme other than the active site. And this changes the tertiary structure of the enzyme / the shape of the active site itself. So the active site and substrate are no longer complementary. And as you can see, another really pretty graph, if I might say. Let's move on. So now the effect of pH on enzyme-controlled reactions. Changes in pH can lead to the breaking of bonds holding the tertiary structure by disrupting the hydrogen ionic bonds or changing the charge on amino acid. The enzyme / active site loses / changes shape and is said to be denatured. The substrate will not bind / fit to the active site, so there'll be fewer / no enzyme substrate complexes formed. In turn, this decreases the rate of enzyme controlled reactions. Once again, a really cute diagram. Moving on, Let's see the effect of temperature. Increasing the temperature will increase the kinetic energy. This increases the collisions between the enzyme. Oh my gosh, there is a spelling mistake there. Don't mind that. Between the enzyme / active site and the substrate. So more formation of enzyme substrate complexes, which in turn increases the rate of reaction. If the temperature is too low, this would decrease the rate because there is less kinetic energy, so fewer successful collisions between the enzyme / active site and the substrate, so there are fewer / no enzyme substrate complexes formed. And if it's too high, this will also decrease the rate because even though there is an increase in kinetic energy, the enzyme will vibrate more, which in turn, this vibration leads to the breaking of hydrogen bonds in the tertiary structure, which changes the shape of the active site and makes sure that the enzyme is considered denatured. So there are fewer / no enzyme substrate complexes formed. And there's another really cute diagram. And this would be the end of the little revision video on enzymes. I hope this was really helpful. Make sure to like, comment and subscribe. And if you have any tips that you want to share with any other students, make sure you comment those down below. And also, I will put it on the screen now that I have made a booklet on the whole biological molecules, and I intend to do it for each topic within the AQA A-level biology course, and hopefully I will get onto the OCRA A-level course. And if you want to purchase this will be down below in my description. I did try and make it as affordable as possible, and also other affordable resources you can find are on TikTok as well. So with further ado, happy revision.